Hello everyone, welcome to 12th Biology Maharashtra State Board. I am Mr. Praveen Kumar Chauhan from GE Society's Sir Dr. M.S. Gosavi Junior College of Science and Commerce, Nasik. Today we will talk about the unit number 3rd, Human Physiology. Our chapter is Control and Coordination. Subtopic is Neural Control and Coordination, and in that we are studying the basic concepts and evolution of nervous system. This is our first lecture. Let us see content at a glance. In this, we are studying the introduction to control and coordination, then evolution of nervous system, response to stimuli in sponges, nervous system in Hydra, nervous system in Planaria, neural tissues, structure of nerve cell, types of nerve cells, types of neuroglial cells and their functions. Friends, the control and coordination is the most important process among all the different life processes. So let us see the introduction to the control and coordination. As we know, unicellular organisms have a simplest organization of their body and life processes. However, multicellular organization of the body system requires both control over their life processes as well as coordination between the various systems in order to maintain homeostasis of the organism. So homeostasis means the maintenance of internal environment of the body, including different hormones and number of different chemical coordinations of the body. Plant and animals both show a control and coordination mechanism, but here the mechanism in different way in plants and in animals. In plants, this is done by sending chemical signals and bringing about various types of movements. For example, phototropic movement, chemotactic movement, thigmotactic movement. So all these three types of movements are observed by plants and they are considered as a part of coordination or the control in case of plants. However, in animals, especially the higher vertebrates, shows gradual increase in the complexity of their control and coordination by giving both electrical and chemical signals. So let us see the evolution of nervous system. During the course of evolution, the nervous system gradually evolved from diffused neural system to a centralized nervous system. Nervous developed or neurons developed as highly specialized cells that generate electrical signals and transmit information. Gradual expansion of neurons system into properly organized system took place. The evolution of human brain has been the most significant event in the evolution of life. The brain increased in size and in functioning over the years. The sense organs detect changes in the environment and send it in the form of impulse to the brain. The brain interprets and responds to the signals if necessary. This has led to centralization of various sense organs assigning in coordination between internal and external environment. The evolution of complex networking system has led to the efficient transmission of signals from one part for organ to another organ of the body. Let us see the nervous system in sponges. As we know, the sponges are the very simplest organisms belongs to the phylum porifera, as they are just shows minimal cellular organization without forming tissues and organ system. So they are the organisms which cell aggregate body plan, and they have number of different types of cells with minimal division of labor. So they have response to stimulus in different ways. So sponges are the simplest multicellular organisms without tissue and organ system organization. Sponges shows cell aggregate body plan, and different cells have minimal division of labor. This led to the specialization of cells for various activity like digestion, respiration, excretion, and others. So here you can see the body of sponge. Here the big structure at the anterior region, that is called as osculum, and number of pores are there called as ostia. Here this particular cavity, that is central canal, is lined by number of cells including the amoebocytes and choanocyte cells, or also called as collar cells. So these cells are efficient, or these cells has ability to digest the food material, and similarly number of cells have different ability. So here neurons are absent, but still sponges respond to external environment. Pinacocytes of outer body wall are highly contractile cells. These can increase or decrease overall surface area of the sponge body. Let us see the nervous system in case of Hydra. It is again the lower organism. It is slightly evolved than that of the sponges. So Hydra is a cnidarian, shows the diffuse nervous system, which is the most primitive nervous system. The cnidarians are thus the first animal group showing true simple nervous system. It consists of sensory cells and nerve cells along with their fibers. The nerve cells are distributed throughout the body and interconnected to each other by synapses. So these are the organisms with neurons, and they are the first organisms, those who having the synapse for communication between the two adjacent nerve cells. So due to formation of these intercommunicating structures, the entire nervous system is in the form of net-like structures and called as nerve net. There are two nerve nets, both in the mesoglea, one connected towards the epidermis and second towards the gastrodermis. Sensory cells are scattered in the body wall and tentacles, but sense organs are absent. Hydra has neurons but no brain, and neurons are polar type. Impulse shows no polarity or direction. Thus, in Hydra, activation of sensory cell can happen at any point, and from this point impulse can be carried throughout the body in any direction, thus bringing movement of the body or tentacles. So here you can see, due to scattered nervous system, or there is no any kind of central control of the nervous system, here the nerve impulse can generate it at any point and shows its distribution towards the any direction. So this is what the nervous system in case of Hydra, and this one is the first organism who shows a true nervous system with the nerve cells. So here in this picture you can see the structure of real Hydra, and here you can see nerve cells forming nerve net. Here the mouth opening around to the mouth, here you can see tentacles, and all over the body nerve cells are interconnected with the help of poorly developed synapse, but the nerve cells are scattered all over the body and shows the nerve net. Let us see the nervous system in case of planaria. Planaria is slightly evolved organism than that of the Hydra because it belongs to the phylum Platyhelminthes. As we know, the Planaria is the flatworm belonging to the phylum Platyhelminthes. The Planaria shows beginning of the cephalization. So as we observed in the previous group of organism, that is the Hydra, which shows scattered nervous system or the nerve net type of nervous system. There is no any kind of centralized control in case of Hydra, but this one is the organism, that is Planaria is the organism which shows beginning of the cephalization. So here slightly centralized control observed in the nervous system. It is the most primitive animal with central nervous system located on the ventral side of body. So initially, as the organisms start their evolution, they showed their nervous system on the ventral side of the body. Mass of anterior cerebral ganglion appearing like an inverted U-shaped brain. Cerebral ganglia arise 9 branches towards the outer side. Ventrally from below, the ganglia arise a pair of ventral nerve cord, that is VNC. Ventral nerve cord are interconnected to each other by transverse nerves or commissures, which gives ladder-like appearance to the nervous system in case of planaria. The peripheral nerves plexus arising laterally from the VNC, that is ventral nerve cord. The PNS includes sensory cells arranged in the lateral cords in the body. A pair of photosensory cells called as eye spots are located on the dorsal side of the brain in anterior region. So here you can see the structure of planaria and the arrangement of nerves, how they forms the ladder-like structure in the entire body. Here slightly encephalization observed in this particular organism at the anterior region, and here eye spots are there which are photosensory structures. So it is said that eye spots are the first eyes in this world. Then here the brain and shows nerve cords on the two lateral sides. The nerve cords or the two lateral nerve cords are interconnected by means of transverse nerves. So here the structure of the body of this planaria, here the mouth, then pharynx, gastrovascular cavity, and on the anterior side you can see the eye spots, and eye spots are present on the structure that is called as brain of the planaria, or also called as ganglia, and these are the two lateral nerve cords. Let us see the introduction to the neural tissue. As the nervous system is the most important system in our body for the survival of an organism, its tissue is somewhat modified tissue as compared to rest of the body tissues, and has its own speciality of conduction of nerve impulse as well as storage of information, which can be interpreted in different ways. So here the structure of nervous tissue or neural tissue. The neural tissue consists primarily of two types of cells, the neurons and neuroglia or glial cells. Nerve is a bundle of axon. The word nerve is used for bundle of axons outside the central nervous system. The term tract is used inside the CNS for nerve. The nerve may be sensory or motor or mixed type. All these, along with the nervous organs, make up a nervous system of the higher animals and bring about coordination and control of various activities of the body. The control and coordination is done through the receptors, which bring in sensory input towards the central nervous system. Processing is carried out in the CNS and then throughout the motor commands. The response is sent out the nerve arising from the cytons of the CNS travel throughout the body, transmitting the nerve impulse to or from the CNS. So let us see, this is the nervous tissue, and these are different types of nerve cells on the basis of the cyton and its position and the structure of axons. So the nerve cell, the bigger region of the nerve cell is called as the cyton or actual cell body, in which the central big nucleus, then in the cytoplasm Nissel's granules are there, then cell body, then on the cyton small structures or somewhat filamentous structures observed here. These are called as conical structures, these are called as dendrites. Then here elongation you can see on one side, that is called as axon, which is surrounded by the Schwann cells and myelin sheath. Here myelin sheath is somewhat interrupted at certain region and forms a node-like region called as nodes of Ranvier. At the tip of this axon, that is axon terminal, here synaptic knobs are observed. Structure of typical nerve cell. Neurons or nerve cell, these are the structural and functional unit of nervous system. Each multipolar neuron has three parts: cytons, cell body, cytons that is cell body, dendrons and axons. So let us see the cyton. The cytons are generally found inside the brain, spinal cord and ganglia. The cytons has a distinct central nucleus with a nucleolus and nucleoplasm. It is the most active part of the nervous system. Small groups of cell bodies inside the white matter of brain are called as basal nuclei. Clear film of the cytoplasm surrounds the nucleus, around which there are neurofibrils. Nissel's granules and other cells or other cell organelles are also found in the cytoplasm. As we know, the Nissel's granules are the riboprotein components which plays an important role in the synthesis of an enzyme required for the formation of neurotransmitter. The next, dendron. Dendrons are many small conical processes arising from the cytons. Dendrons transmit message towards the cyton. These are highly branched into fine dendrites. Nissel's granules and neurofibrils both can be seen at the base of dendrons. Structure of nerve cell. So as we observe the nerve cell, here the dendrites, as these are the conical structures or highly branched structures, and dendrites are nothing but the modifications of plasma membrane of nerve cell. So here this is what the entire nerve cell, its structure, its unique structure is due to the modification of its plasma membrane. As the plasma membrane has different structures, it means that there is somewhat cytoskeletal elements which helps in maintaining such kind of structure. Here the big size nucleus with nucleolus. Nucleus granules are there. Then cell body, Schwann cells, axon, myelin sheath, nodes of Ranvier, etc. So here the ultrastructure of the nerve cell, which shows different cell organelles as that appears in the normal animal cell. Axon, it is a single long, usually unbranched process arising from the cyton at the axon hillock. It consists of a bundle of neurofibril. The axons carry the message away from the cytons. Terminally, the axon gives out branches called telodendrons. The axon may give out lateral branches are called as collaterals. The terminal branches attach to a muscle, gland, skin, or telodendrite of another neuron. Neurofibril play an important role in transmission of nerve impulse. The nerve can be covered only by neurolemma in the non-modulated nerves, while in modulated nerves it is covered by both medullary sheath and on the outside by neurolemma. So neurolemma is nothing but the plasma membrane of nerve cell, and myelin sheath, it is somewhat insulated structure made by the lipid. Conduction of impulse by the modulated nerve is 50 times faster than in the non-modulated nerves. This is because of there is minimum wastage, or even there is no wastage of the nerve impulse or whatever action potential is there through the modulated nerves. Whereas in non-modulated nerves, there is some sort of wastage of that action potential which is generated, so that its transmission is quite slow than that of the modulated nerves. Connective tissue coverings of nerve are endoneurium, perineurium, and epineurium. So connective tissues are also contributed in the nervous system, and they covers the nerves or the nervous system elements, including endoneurium, perineurium, and epineurium. Types of nerve cells: unipolar nerves, bipolar nerves, pseudo-unipolar nerves, and multipolar nerves. So what is unipolar nerve? Unipolar nerve means what? Here cell body at one side and the axon is there in single axon, that is unipolar nerve. Bipolar nerve means what? Here you can see the sensory receptor at one end, then cyton at the middle, and here both the sides of the cyton shows the axon region, hence it is called as bipolar nerve. Pseudo-unipolar nerve, it is the structure in which the axon is almost single but has a short branch which keep the cyton slightly away from the center, and hence it is called as pseudo-unipolar nerve. Multipolar nerve, as here its structure is more or less similar like that of the unipolar, but the most important difference is your cyton has multiple dendrites and even axon has telodendrites, so that's why multiple poles are there, hence called as multiple or multipolar neuron. Types of neuroglia. As the neuroglial cells are the supportive cells in the nervous system, they are of different types: ependymal cells, then oligodendrocytes, satellite cells, astrocytes, microglia, Schwann cells. Let us see the neuroglia. Neuroglial cells are much greater in number than the neurons. The different types of neuroglial cells are oligodendrocytes, microglia, or brain macrophages, astrocytes, satellite cells, ependymal cells, and Schwann cells. These cells are supporting cells of the central nervous system and peripheral nervous system. Most of the supporting cells of the nervous system are divided from ectoderm. So as we know, there are different systems, shows different origin or different germ layer origin in our body. Here the nervous system has ectodermal origin, and whatever nerve cells are there, they are derived from the ectoderm, or neuroglial cells also derived from the ectoderm. Types of neuroglial cells. So first type, that is oligodendrocytes. They are found in central nervous system. These cells have few branches. Oligodendrocytes mainly from myelin sheath, or they forms myelin sheath around the central axon, which forms the white matter of the CNS. So as we observe that the white matter in the CNS is due to the appearance of the myelin sheath, or appearance of the axon which is covered by myelin sheath, and this is the major role of oligodendrocytes. So ultimately, oligodendrocytes are responsible for the appearance of white matter in the CNS. Microglia or brain macrophages. As the term macrophages, these are the cells which has some sort of ability of phagocytosis, and these are the small size cells found in CNS with two branches. They are derived from the monocytes. As we know, the monocytes are the cells that found in case of blood, or they are the leukocytes, and acts as macrophages. So they reached in the site of injury. Dead neurons and cell debris in the CNS are cleared by the macrophages, or the microglial cells. They mediate immune response in CNS. Next type, astrocytes. These are star-shaped and the most abundant glial cells of the central nervous system. They have very good role in the brain, that is secretion and absorption of neurotransmitter and maintenance of blood brain barrier, that is BBB. They regulate the transmission of electrical impulse with the brain. Ependymal cells, they are found in CNS. The cells from single layer of squamous epithelium, columnar cells of ciliated epithelial cells lining the ventricles of brain cavities and central canal of the spinal cord. They are mainly responsible for production of CSF and also for circulation of CSF, as they have the cilia, so that circulation of CSF made easy. CSF in brain and ventricles and even central canal. Schwann cells. These are the most abundant glial cells of the PNS. They produce myelin sheath around the mediated nerves of the peripheral nervous system, that is PNS. Satellite cell. These cells are found in PNS. They support the function of neurons. Thank you very much.